I want to do a very brief introduction as to who Bruker is and what we do. So Bruker is a German organization, and with 7,500 employees growing all the time with 14 manufacturing sites across the world. We have 1,200 employees dedicated to research and development, and about 9% of our overall revenue goes back into R&D every single year. So a huge research team, and we currently have around 4,000 patents pending or granted, so a real innovation organization. So Bruker are really known for instrumentation across many different industry sectors. I don't know how many of you have heard of the name Bruker, but so for instance, if you've ever seen Fake or Fortune, the BBC programme with Fiona Bruce, at the start there's somebody firing a gun at some painting to see what pigments is made up of. That is a Bruker instrument, for instance. So lots of different applications for Bruker instrumentation. So today, the sector we're going to concentrate on is the microbiology division, microbiology and diagnostics, and a particular instrument called the Multibiotype. So just to give a bit of a timeline, so Bruker was formed in 1960 in Germany, introduced the first Malditof mass spectrometer, which is what we're going to talk about today, in the 90s. And really it was the introduction and the launch of the Multibiotype IVD system in 2009, where it really did revolutionize microbiology. And today we have over 4,000 instruments placed globally, so market leaders by a long way in the UK, for instance. So what happened before Malditof? So traditionally, when you send a patient sample off to the laboratory, whether that be a wound swab or a blood culture, which is what we're going to focus on today, what you need to do first of all is determine, is there something in that sample? Is there a real infection? Because as we talked about before, the sepsis, you don't get sepsis without an infection, but also to understand what that causative organism is. So what is causing that infection? That could be a bacteria, a yeast, a fungi. And prior to Malditof coming in, there were many different manual and semi-automated methods which could take many hours or even months, incredibly labor-intensive and sometimes not very accurate. So with the introduction of the Multibiotype, the time to identification of the organisms was reduced really just down to 1 minute. And it employs lots of high-tech gadgetry. So essentially what you do is you put your sample onto a target plate, you fire a laser at that target, and then it is able to identify proteins from those organisms, and depending on the mass of those proteins, it gives you a fingerprint of exactly what those organisms are. So this is proteomics in action, and I think anything with a laser in must be good. So there you go. So when we focus on the sepsis pathway and the blood culture pathway, as you know, you would take blood cultures if you suspect sepsis, send it off to the laboratory, you incubate the bottle in a blood culture incubator, and then normally what you would then do is take some of that blood culture, put it onto an agar plate, grow it for another 24 hours potentially, and then run an ID. With the introduction of Sepsityper, what we're able to do is reduce that time to identification from 1 day down to about 20 minutes. Massively, obviously, improving the information that's given back to the clinicians on the ward. So apologies for a bit of a busy slide, but this sort of demonstrates how busy Bruker are at the moment, in that they're moving away from just being known as an identification company and an instrument company, with the introduction of new methods through acquisition and through research and development, starting from the patient journey with pre-admission screening, using molecular techniques for things like CPE carriage, through the blood culture pathway, where they're also directly from the Sepsityper sample able to do some antimicrobial resistance testing using the STAR kits, which Francis will talk you through, right through to the following day with confirmatory testing. But really it's the MBT Fast that we're currently really excited about, which by the end of this year will be launching direct on Moldy antibiotic susceptibility testing. So again, using Sepsityper as the starting point for this test. So a lot of exciting times ahead for Brooker. So that's the brief intro. So now over to Francis.
Okay. Hi everyone, so my name is Francis and I'm the laboratory manager for microbiology at Great Ormond Street Hospital. I'm also a clinical scientist, and so what I'm going to be presenting is basically utilising rapid diagnostics and new methods to quickly diagnose sepsis in the laboratory. So this is just a bit of what I'll be talking through, so just a bit of the agenda of the talk. And I'll basically start off with where I work. So I currently work at Great Ormond Street Hospital. I've been there for nearly 9 years. It'll be 10 years next year, so I've been there for quite a while. But before that, I was working at a district general hospital for 3 years. So you've all probably heard of Great Ormond Street Hospital. It's a very unique place. We get referred a number of patients for rare diseases, but it's also specialists for bone marrow transplant, oncology patients that are very ill. But it's not just patients from the UK, it's also patients internationally. So we get patients from Saudi Arabia or patients with specialist conditions where they need to see a surgeon or specialist treatment and need to be monitored. And so they do get referred to us to see the specialists. This is a nice picture from the London Olympics 2012, which is actually when I just started. I always like to talk about the lab as a scientist, and I think it plays a crucial part in diagnostics. So a little bit of the background of the microbiology laboratory at Great Ormond Street Hospital. So we do about 125,000 tests per year, which may not sound like much compared to other labs, but again, our lab is on site, which we're very fortunate about, and it actually focuses on our patients. We have 40 healthcare scientists, so that's a mixture of different groups, which include biomedical scientists, clinical scientists, 11 support staff, so that's medical laboratory assistants who have a crucial role of actually checking when the sample comes into the laboratory, that actually everything's labelled correctly, it's the right patient, the integrity is correct, we can perform the test, otherwise we need to contact the service user. In the micro lab, we basically do bacterial identification, so when we find a pathogen that's significant, we ID it, and then we carry out antibiotic sensitivity testing. And that's just so that we can actually report to the clinicians, to pharmacies, so they actually know what they want to treat with, what's appropriate. So going back to when I first started, when I actually got to Great Ormond Street Hospital, I was like, oh wow, amazing place. But actually, it was quite far back in time in terms of diagnostics and tests. So they use a lot of manual tests. They use something called an analytical profile index, which is a biochemical test. And so when you're waiting for an ID for an organism, it would take 24 hours. And that's quite crucial, knowing that ID, because bacteria is quite significant in that they can be resistant to certain antibiotics, they can be sensitive to different antibiotics as well. And so when you have that ID, that immediately changes how you manage that patient treatment, etc. So, there was always a need to kind of improve things within our hospital, and particularly when you've got a patient population where they're, children, we need to kind of treat them very quickly, rare diseases, you want to try and get a result out as quickly as possible to improve that treatment and management. So basically in 2012, we decided to kind of bring in the Malditof because looking through the literature and a couple of other hospitals that used it, they actually showed the benefit of it, which actually was to actually reduce the time to identification, which is what we want to do. And actually it also changes our workflow in the laboratory. So actually, when you know your ID so basically when you get a patient sample in the lab, we culture it onto agar, goes into an incubator. When it comes out, we look at that culture, and if you can ID it straight away, that means you can set up the right antibiotic sensitivity straight away so that you can get that result out much more quicker. And we can also let the medics know, actually, this patient's got this, and they can give some kind of preliminary treatment. But it has improved the workflow in the laboratory because actually having that information more quickly means that you can actually do things more quickly and get those results out more quickly. So we're wanting to improve turnaround time as well. This is our Moldy, and it's named Bonnie because the day that we actually got approval for it from funding, it was the day that Bonnie Tyler was actually at the Eurovision Song Contest. So. Okay, so about the implementation of Multibiotype. So this is kind of the same with any diagnostic instrument. Before you bring something into the lab, you need to check that it's fit for purpose. So we basically ran a number of known samples through it to check actually we're getting the correct ID, that it is, what we put through is what it says it is. Moldy is brilliant, but there can be little bits that you need to just check you're getting the right ID, and so that's why we need to be sure before we're putting out patient results that it's doing what it says on the tin. We confirm these with control organisms, so this is bacteria which are known strains or ones that we've confirmed by molecular techniques in the laboratory. We used to use a Vitek, which is another diagnostic instrument for ID, and compare that to that as well. And actually, we were very happy with the result, and so we were happy to move over and sign off that validation to implement the Malditof into the laboratory. So yeah, I mean, as I've said, and kind of harping on it, has improved things when it came into the lab, because the lab utilised it so much for ID and was so reliant on it for getting those IDs to help, and it reduced cost as well. So in the laboratory, we use a number of chemical tests, etc., and actually we could stop using those just because we were using the Malditof, and actually it was cheaper as well to use the Malditof as well. So, okay, sepsis. So sepsis results from an overreaction of the body's immune system to infection, where our own tissues and organs are attacked. It can lead to septic shock, multi-organ failure. So this is kind of a nice description, and it's a crisis everywhere. 30 million people worldwide every year suffer from it, and it's quite severe. So we want to try and tackle it as quickly as possible. So sepsis is a serious clinical condition, about 123,000 cases per year, maybe more now. And obviously there is local guidance, nice guidance in terms of using the Sepsis 6 for quick treatment. There are other things you can do in the laboratory. So within GOSH, we've actually done a lot of kind of quality improvement work to get blood culture to the laboratory as quickly as possible through comms, so communication, telling the wards, don't just get a porter to bring it. If you can, send it through the pneumatic chute. Once it comes to the lab, we tell the lab staff priority, get that on the machine as soon as possible so that you incubate it and get it. If it's positive, we can get that working, basically. Okay, so I've talked about using Malditof for IDing bacteria from cultures, but actually it has a lot of other things that it can do. So we actually decided to then implement the Sepsityper for blood cultures. So what the Sepsityper is, it's this little kit here, and it reduces our workflow in the laboratory. So what happens is when a blood culture comes to the laboratory, we put it on the instrument. It's incubated for about 7 to 9 hours. If it's positive, it flags up at different times depending on what organism is present. When it flags positive, the biomedical scientist will take it off the instrument and work it up. So you usually do what's called a gram film, so it's a little film, and you look at the morphology, but you can't tell what ID that bacteria is just from that, because it could be anything. If it's what's known as a gram-negative rod, it could be E. coli, Klebsiella, Pseudomonas, anything. This basically reduces the time and actually gives you an ID from the moment it actually flags up positive. So you've got that ID. So, for example, the blood culture flags up positive, you do a Sepsityper and you've got that ID, and then within our laboratory, they actually ring it to the ward and say, oh, we've had a blood culture flag up positive on this patient, this is what's actually been found. This is a preliminary ID which we will confirm, but this is what we've got. So it's quite an improvement. So this is just a little bit of the workflow that I've kind of already described. So obviously there's kind of the blood collection. It goes on to a blood culture system where it's incubated and rocked. And basically that will, if there's any pathogens present, they will basically grow. Again, in our laboratory, if it flags up positive, we then do a Sepsityper, but only if it's a new known blood culture. So if it was like the patient had one positive within the last week, we would probably leave it and rely on the gram stain unless it was something different. But all new patients who've had a new blood culture flag up positive, we will do this Sepsityper to get that preliminary ID, and then that goes on to the Malditof. So again, it's getting a positive result, an ID or preliminary ID, we get that within 15 to 20 minutes of it flagging positive, so that's quite significant. So just a bit about the principle. So the MBT Sepsityper IVD kit selectively destroys the blood cells and enriches microorganisms from a positively flagged blood culture. And the resulting cell extracts and pellets are suitable for analysis using the Multibiotype. The MBT Sepsityper IVD kit enables a high probable identification of the relevant microorganism species within 15 to 20 minutes of using the Multibiotype. Okay, so I'm just going to go through some case studies of patients that have actually benefited from the Malditof. So firstly, case study 1. So this is a patient, a neutropenic patient. They basically had a raised CRP. They suspected there may be sepsis, so then they took some blood cultures of the patient and brought it to the laboratory as quickly as possible. They were put on the system, and then they flagged up positive. So I'll kind of look at how we would have done it previously to when Maldi had actually come in. So our old way, if the bottles had flagged up positive, we would have done a gram film. That gram film, so in this gram film, it's gram-negative rods. So that could be a number of different organisms. It could be E. coli, Klebsiella, Pseudomonas. Looking at the patient history, there's not actually any record of any kind of colonization, because we do a lot of screening for our neutropenic patients. Because they're more septic, we like to know what they're carrying, because again, they can get septic more quickly. And so just having just that information that it's a gram-negative rod, what pharmacy would do is then actually prescribe the patient meropenem. So now, let's go to like using the Sepsityper. So for this case study, we actually did use the Sepsityper. So it flagged up positive. They did an ID, and the ID of the organism was Stenotrophomonas multifida. So I don't know if you know about this bug, but basically it's resistant to meropenem. So, having the ID, suddenly you've changed something immediately. So then actually they could actually implement correct antimicrobial stewardship, and so the patient was put on a high dose of trimethoprim sulfide trioxole. So basically it benefited them having that change, because they would have still been ill, but actually having that result, you can act quite quickly, and it's also improving antibiotic prescribing, which is something quite key in the future as well. So just a second case study. So a patient in our intensive care unit, they were ill and they had a number of different samples taken, brought to the laboratory, and one of them was blood cultures which had flagged up positive. So again, our old method, we would look at a gram film, and in this gram film we saw gram-positive cocci. So usually, depending on the morphology, if it's in a chain, if it's looks like a bunch of grapes, it might be a staph, it might be a strep, could be a number of things. So because it's gram-positive cocci, they initially started on vancomycin. So now, when we use the Sepsityper, after it flagged up positive, they actually had a Staphylococcus aureus. And so this actually, again, changed the management for the patient. So alerted we got the result, we phoned it straight to the intensive care unit to let them know, bacteremia, prompting early line removal of the driveline, therefore changing therapy and hygiene management of the patient. Okay, so further development. So, obviously, introducing Malditof, introducing Sepsityper were really, big changes to our laboratory, but we always are trying to drive to still improve. We got a new blood culture system, and while we were validating that for use for our new blood cultures, we also trialled a new rapid shortened procedure. So for blood cultures that had flagged up positive and following the Sepsityper method, we actually removed a step just to reduce it even further. And again, it's to simplify the workload for the laboratory. So in my lab, basically, we cover the lab 24/7, so there's always someone in the lab. It's only one person, so you can imagine if you're in the lab alone, the phone's going off, blood culture machine's going off, and you've got lots to do, and you're trying to focus. So we were trying to reduce the workflow and stream just so that actually it was easier. They could get that report more quickly, as well as kind of doing other duties too. And the main thing about this reduced, shortened method is that it's less hands-on time. So while we were validating our blood culture instrument, we did about 80 blood culture bottles, which we spiked with organisms we have in the lab and tested. They all flagged up positive. So we tested an array of different organisms that are listed here. And then basically what we did is we followed a Sepsityper method using the normal Sepsityper method as prescribed by the protocol, but then also did a reduced one, so using the same workflow, but kind of reducing a step, so you're just actually using the pellet directly rather than doing some extra washes. So this is kind of the workflow as it goes. So your blood culture flags up positive. So you harvest 1 ml of the blood culture into an Eppendorf tube, which is that little plastic tube there. And you add a lysis buffer, which actually breaks down the red blood cells. You centrifuge, so it's basically spinning it down. And if there's bacteria present, it will basically concentrate that organism into a little pellet. And then you add a washing buffer, again, just to clear it, and then centrifuge it again. Now, at this point, there would be further washing steps, but actually those two washing steps have already done enough clearing, and that pellet is your organism, your bacteria concentrated already. So what we would do is actually touch that pellet and put it directly onto what we call a target plate, and we'd fix it with formic acid, which would break the bacteria cell wall down. We would add matrix to it, which will hold it in place, and then it can go straight on the Malditof. Whereas before, if we carried on the full Sepsityper protocol, again, you're adding a couple more washes, and that takes a bit more time. It's with ethanol, etc. So we just tried reducing it and trying to compare the two different methods. So this is just the results, basically. And you can see the top line is the full extraction, so that's the protocol as written. And then below is the rapid workflow. So the full extraction has done better in the sense that it's 97%. We've got good IDs, but then actually rapid workflow hasn't done too bad. It's 95%. But then again, if you wouldn't get an ID, you would go back to your full method, or you kind of wait basically until the next day because you couldn't get an ID. So we always try our best. But I think the significant part of this data we need to look at is that actually full extraction is about 20 to 30 minutes hands-on time. And then the rapid workflow is 10 to 15 minutes. So we're always trying to work to reduce the time to identification. Okay, so benefits of the MBT Sepsityper. So again, as I've said already, reduce hands-on time. The rapid workflow is showing reduced hands-on time to 10 to 15 minutes from the previous 20 to 30 minutes. Reduced time to diagnosis. So compared to previous methods using gram film, the MBT Sepsityper IVD workflow has reduced the time to diagnosis by up to 24 hours. And I think the key thing from the case studies that we've seen is improved antimicrobial stewardship, which is something, we've all been hit by COVID and discussion about COVID. I've heard it enough in the lab. But actually, I think other things will be on the agenda. And I think, antibiotic resistance is one of the things that are creeping up. And so if, we are always trying to work in the lab, but not just the lab, I think on the wards, within infection control and pharmacy to improve prescribing behaviours. And I think when you're doing a case study or working up a patient, having all the information is quite key to improve those behaviours. So I've said about enough developments with Malditof in our laboratory that we've done already, but we're still planning more, and I think there'll be more ahead for us. So I've talked about resistant organisms. So we're an international hospital. We get patients from all over the world that are referred for specialist treatment. And so what comes with that is because each country is quite different in terms of their prescribing, there are some countries which are more prevalent for having resistant organisms. So that is quite scary in the sense that bacteria, the way that they work, they're very clever and they can transmit different genes to other organisms so that they acquire those resistant mechanisms, which is quite scary. And so for us, one of the key things, as well as IDing pathogens, is actually knowing this is this pathogen. It's actually a very resistant, we saw about that's Trophomonas, but there are other bugs that have got resistant mechanisms. And actually IDing those and knowing actually it's a resistant bug improves the treatment and management quite quickly. So something that we've been looking at is the MBT Carbostar, which is this kit here. And this is basically looking at the bacteria and trying to basically identify if there's a resistant mechanism present within it, and having that result a bit more quickly. So again, the way that we work things up, we basically, in our laboratory, we do our ID and culture, and we do antibiotic sensitivity by what's called disk diffusion testing, which is another 18 to 24 hours. But actually using this method, we're working at the validation at the moment, is hoping to reduce that even more greatly so we can know that result more quickly. And that's more so for infection control, so that they, in terms of managing a patient and stopping, transmission to other patients, they can kind of do that. So I guess the key things about transmission. So the kit allows a quick detection of carbapenemase by enzymatic activity by a mass spectrometry test on the Malditof. So as I've said already, we currently are using a lot of hands-on tests. So we do these things called MIC strips. So on that is an antibiotic which is concentrated at different levels. At the top number where the letter is, that's basically the highest concentration. So you can see that organism on there is resistant to both of those antibiotics because there's no zone of inhibition basically. So that antibiotic's not working, which is quite scary. So we do tests like that in the laboratory, MIC tests, or confirmatory disk tests and measuring zones to check those different resistant patterns. But actually trying to implement Malditof would actually be quite beneficial to us having that and improving things within our workflow. So as part of looking at Malditof, there are other tests we're looking at. We're trying to basically make a workflow in terms of looking at CPE so that we can identify things quickly and accurately too. So the new CPE algorithm, so it's an improved diagnostic workflow so that a rapid CPE result can be obtained based by the MBT Sepsityper IVD and the MBT Star BL result. This will be done by designing basically a new algorithm which will give quicker detection of CPE and negative results. So this is kind of testing within our laboratory. So obviously the scientist staff, when they're looking, they know that they're following the correct workflow and know that basically an organism needs to go through a certain stream to get that result out quickly enough. And quicker detection to improve infection control procedures, as I've mentioned previously, and stop transmission. So other things that we're basically looking to kind of develop, so you've all probably heard of MRSA. And at the moment, so where I'm working, we grow it on culture, and then we do a number of tests to ID it. So we, Malditof can tell you it's a Staph aureus, but it won't tell you actually it's got the resistant mechanism that makes it an MRSA. But actually we're working to develop that and build that in our laboratory because we have to wait 24 hours to get that result to confirm it's an MRSA by looking at a cefoxitin MIC or disk test. But actually, if we can get it at the same time that we ID, that will, again, be a cost saving for the lab, but again, help with infection control and transmission for patients. So just going to talk about precision AMR, so just a little sideline. But this is a project between GOSH, Great Ormond Street, and UCL. So it's a government-funded project, and basically it's been funded £2 million for several different laboratory research projects to basically look at improving AMR. So bringing in rapid diagnostic tests, such as utilising Malditof or other kind of tests as well, and basically working to try and firstly improve antibiotic prescribing and the time for identification of tests and resistant mechanisms. So just to kind of like summarise, has the Malditof benefited GOSH microbiology? Yes, greatly. The workflow, but not just for the laboratory. I think in terms of getting results out to patients, changing that treatment and management. So kind of just to conclude and summarise, so it has improved antimicrobial prescribing based on MBT sepsis type IVD rapid workflow. Identification results from positive blood cultures can now be delivered up to 48 hours faster to the clinician, resulting in faster change of therapy. Improved infection control by MBT sepsis type IVD, so by changing bed management of patients depending on the identification of the organism that's found. Cost savings for the trust by reducing the use of antibiotics. Also, quicker diagnosis means quicker action for adopted treatment, so patients can stay can be reduced. I think that's really critical for our patients at GOSH, because again, we've got kids, and it's so horrible if you've got a little child, and you're having to isolate them in a room, and they get very upset. So not just benefiting all patients, but I think thinking of the patient directly as a child. So yeah, overall patient benefit. And that's it from me. Thank you for your attention.